Hello, everyone. Welcome to week 2 of the 1124 lab. Again, I'm Ed. I'll be your TA for the rest of the semester. And if you need to get in touch with me, my email is higginse@ou.edu. So let's get started on this week's lab. This week, we're going to be doing an investigation into the macromolecule content in different kinds of foods. So I'm going to be giving you some tips on how to make the most of this week's lab. So during lab this week, we're going to identify and describe various types of macromolecules, determine the macromolecules in common foods, and then you all are going to write an experiment summary and then comment on Flipgrid, and I'll get more into that later today. So first, we're going to talk about the different types of macromolecules. First macromolecule that we're going to dig into are carbohydrates. And so the basic building blocks of carbohydrates are going to be sugars. So when you have just one sugar molecule, it's a monosaccharide, mono meaning one. And when you combine two of them in different combinations, you'll get a disaccharide. So the monosaccharides we're going to talk about are ribose, glucose, and fructose. They're all similar looking molecules, but the amount of carbon and hydrogen composing them is going to be different, and that's what matters when you combine them. So when you combine, for example, glucose and fructose together, you get sucrose. And so these basic sugars, either monosaccharides or disaccharides, are going to be used as cellular food sources, and they're very quick food sources. They're easy to break down because it's just a couple of molecules. And when you have them present in food and you use Benedict's reagent to test for them, it'll turn bright orange or red. So just keep in mind that when you're using Benedict's, the presence of sugar, the basic building block, is indicated when it turns red. So then next we're going to have the starches. And so starches are a polymer of many glucose molecules. And so a polymer, poly means multiple. So when you have more than two, you're going to end up with starch. And so this is used as both structure in plants and energy storage. And so, if you have too much glucose and you want to keep it for later, you're just going to link them all together and you end up with a starch. And so the presence of a starch, so the bigger molecules that have not been broken down yet, is indicated by iodine. And iodine is going to turn black when there's a lot of starch present. So with carbohydrates, you just need to make sure that you're understanding the difference between the two tests. One is testing for the full molecule, and the other is testing for the molecule when it's been broken down. So just keep that in mind for later. So next we have proteins, which I'm sure a lot of you are familiar with. These are polymers, so these are a lot of amino acids strung together. And these proteins are really cool. They can have very complex structures, and the function of a protein is always determined by the structure. Kind of think of them like puzzle pieces, where the shape that the protein or enzyme is in is critical to what it's going to work with. And so enzymes are what we call these proteins that have active things that they do. And so they'll basically take a chemical reaction that, while it might happen in nature on its own, the enzyme is going to really speed up the process, and it can build things, it can break them down, it's involved in cell structure and transport. And so the presence of proteins is going to be indicated by the biuret reagent, which will turn purple. So you'll see beta chains and alpha chains, and so those are just two of the secondary structures that proteins can take. If you want to learn more about proteins, we can definitely talk about it. They're really cool and they're really important to how cells work. So next we have lipids, which are going to be glycerol and fatty acids. Lipids have the hydrophilic head, and so hydrophilic, hydro meaning water, philic meaning it is attracted to it. So it's going to be attracted to water, and then the hydrophobic tails. And so hydrophobic, hydro, water again, phobic, fear of, sort of, but it's going to repel water. And so this is used for energy storage, again, because you have all of these molecules that are strung together in those fatty acid chains. And it's also used for membranes. So the phospholipid bilayer is one of the more important ones, where you have the lipid and then you have the hydrophilic head that has a phosphate group attached to it. And so they sort of line up and they have a bilayer where you have the chain here and the chain here. And so inside the bilayer is hydrophilic, but outside is hydrophobic. Or the other way around, sorry. And so the presence of these lipids is indicated by the Sudan 4 reagent, which turns bright red. And so in your lab manual, you should have the difference between saturated fat and unsaturated fat. So make sure you know the difference and where they come from. Saturated fat is typically going to come from animals, whereas unsaturated fat is going to typically come from plants. So today in the lab, you're going to be watching a video of it because we're doing a virtual lab. But we're going to go through the procedures because even though this is a virtual lab, in the future you may be taking in-person labs and you'll need to know some of the lab safety and precautions and reasons why we do things in lab. So while you are watching an experiment that's already been designed, you definitely need to understand the components that go into designing an experiment and setting it up. So the first most important thing is that you have a control group. So the control groups are how you know that your experiment worked in the first place. I mean, you can run all of these tests, but if you don't have control groups, you're not going to know that your experiment was functional. It could have had things that go wrong. So the positive control only contains the molecule of interest, so you're just seeing how the molecule acts on its own under the conditions that you're running your experiment. And then you have your negative control, which doesn't contain any of the molecule, so then you can see how the reagents that you add into it act on their own. And so you need to compare these to what your experiment does so that you know what typically happens and then what happens when you actually run your experiment. So the experimental groups that we have are the different foods that we're testing: the hot dog, tofu, energy drink, and milk. And so in this case, the variable that we're testing for, that we're interested in, is going to be the macromolecule that's present when you use these different reagents. And the independent variable, the one that we are testing for the dependent variable, is going to be the types of food. So when you write your hypothesis for your lab summary, remember that a hypothesis is a causal explanation. The goal of this is to be testable. It doesn't have to necessarily be supported. You just have to be able to test it and, importantly, refute it if it doesn't make sense, or if it doesn't work, rather. So, for example, Red Bull turns black in the presence of iodine because it contains starch. That would be your hypothesis. And so this you can prove wrong. I mean, if Red Bull does not turn black, then your hypothesis is not supported and you can reject it. And so that's kind of what you want when you design these hypotheses, is you want to be able to prove it wrong if it's not true. So write a hypothesis for each of the 4 foods being tested and then include these in your lab summary. So if you were to do this experiment in person, it's really important to stay organized, and so you need to label your test tubes so that you just keep them straight. This is kind of, when you're doing science and running experiments, organization is going to be one of the most important things that you do. When writing your lab summary, it's going to be really important that you understand the intricacies and differences between these 4 reagents that are going to be used to identify the macromolecules. Make sure that you know what they identify and what color they are supposed to change in the presence of that macromolecule. This is kind of where rote memorization might come in helpful, just to memorize what these do, but then you're going to need to use your critical thinking when you're actually looking at the results of what's going on. So just make sure that before you watch the video of the experiment being done, that you have a pretty good idea of what each of these are testing for. So lab safety, of course, is very important. So some of these reagents can stain and irritate skin, and other reagents that you may use in future labs may do this. So you need to be wearing your appropriate PPE, even if it weren't COVID times. So you're gonna have to wear your goggles and gloves when you handle reagents and test tubes in the future. When you use pipettes or droppers or anything, the amount of reagent that you add is typically going to be very important in these experiments. And so a lot of times when you use pipettes, if you haven't used them before, they'll have markers on the pipette. So the dropper here on the right doesn't have any markers, so that's going to be the number of drops that you add, versus the pipette that might have 3 mils on it, and that would be how much you're going to add. You pull the volume up to the marker. So again, this is just something to keep in mind in the future when you have another lab that you do in person. You will need to understand how the actual tools work. Yeah, you have to plug things in for them to work. So make sure in the future you do that. And yeah, this is about time management. It'll take in the experiment about 20 minutes for the water to get up to temperature, and then you have to be careful that it's hot, of course. As I mentioned, it's going to be hot, so you have to use the tongs to hold things. This sounds very straightforward, but when you're in the lab and actually running everything, it can get a little hectic, and it helps to have the reminders to actually use the lab safety that you will need. This is actually a more important step than it sounds. You need to clean your tubes, ideally before and after you use them, because other people are going to be using the same glassware that you will be using, and if there's any contamination left in their experiment, then you need to get it out, and you want to be courteous and make sure that you're not leaving contamination in the test tube for future. So if you can imagine that you're, have an energy drink in a test tube and there's milk residue in the bottom, whatever the milk has in it is now in your test, and it's going to invalidate the results of your test. So cleaning is just as important as organizing when it comes to lab work. When you have finished all of your trials, give the test tubes a final rinse with DI water. This is just deionized water, it's super clean water, and then you place them upside down so they dry. This is, again, just being courteous to the next person that uses it and making sure that you are leaving things as clean as you find them. So at the end of the experiment, you're going to write the experiment summary. On Canvas, there are resources that are going to give you an idea of what you need to do, and you'll have a rubric there that'll explain what we're looking for. So just make sure that you, before starting the summary, you make sure you know what needs to be done for the summary. And then I will be sending out an email with a link to the Flipgrid. If you haven't made an account already, make sure you make an account, and then I'll give you the access code so that you can write a comment. And so I'll get into the details of that. So as you are watching the video with today's experiment, or this week's experiment, when you have finished collecting your data, make sure that you summarize the data that you recorded in the lab background in one table for you to use in your lab summary. It should just be one table. This table should contain information about which molecules are present in each food sample. Put the independent variable in the leftmost column, and then place the dependent variable across the top row. So independent, dependent, fill in the graph. So some tips to make sure that you do as well as you can. So I've said it before, and I'm going to say it a second time, which means you know it's important. Review the How to Write a Lab Summary document on Canvas, and review the rubric before you write your lab summary. We're going to have the rubric will have exactly what you need to do to get a good grade on it, and so if you just follow the rubric and follow the document, you will be successful in this assignment. The lab summary should be uploaded to Canvas by noon on Friday. I would advise not procrastinating, because if you upload it at 12:30 on Friday after having a lot of trouble throughout the morning, I still can't grade it because the assignment will be closed. A lot of students have been having issues uploading their assignments. Make sure that you're not using a browser app. You should be using the desktop app. On Canvas, there's a link to the OUIT store. Make sure you're downloading the appropriate apps and saving the document and then uploading that document. So this week you also have a Flipgrid assignment, and so in this video you'll follow the link that I send out later today, or Monday rather, if you're not watching this on Monday. In this 1-minute recording, you should have your name, a brief summary of the experiment, hypothesis, and conclusions that you have come up with. I want to hear the most challenging aspect activity and something that you learned as a result of this activity. So I'll be responding to these at some point the week following when you turn it in. And it'll just help us get to know each other and make sure that the experiments are working and that you're getting the most that you can out of it. So that's all I have for you today. Good luck, and be sure to email me or come to my office hours if you have any questions.